The discovery of CRISPR-Cas9 has ushered in a new era of genetic engineering, offering unprecedented power to manipulate the very building blocks of life. This revolutionary technology allows scientists to edit DNA with remarkable precision, akin to using a molecular scalpel to cut and paste genetic information. The implications of this breakthrough are far-reaching, with the potential to transform medicine, agriculture, and even our understanding of human evolution. However, this newfound ability to rewrite the code of life also presents profound ethical challenges. As we stand on the cusp of this genetic revolution, we must proceed with caution and engage in thoughtful deliberation about the potential consequences of wielding such immense power. The ethical considerations surrounding CRISPR are complex and multifaceted, demanding careful consideration from scientists, policymakers, and the public alike. It is imperative that we engage in open and informed dialogue to ensure that this transformative technology is used responsibly for the benefit of humanity. CRISPR-Cas9 has rapidly become an indispensable tool in the scientific toolbox, revolutionizing various fields with its versatility and precision. In medicine, CRISPR holds immense promise for treating genetic diseases, offering the potential to cure inherited disorders that were once considered incurable. Scientists are exploring its use in correcting genetic defects that cause diseases like cystic fibrosis, sickle cell anemia, and Huntington's disease. Beyond treating inherited disorders, CRISPR also shows potential in combating infectious diseases. Scientists are investigating its use in developing new antiviral therapies and engineering mosquitoes to resist malaria transmission. In agriculture, CRISPR offers the potential to enhance crop yields, increase nutritional value, and develop crops resistant to pests and diseases. The applications of CRISPR extend beyond healthcare and agriculture, with researchers exploring its use in bioremediation, biofuel production, and even in the de-extinction of extinct species. The possibilities seem endless, making CRISPR a truly transformative technology with the potential to reshape our world. While CRISPR's potential benefits are undeniable, its application in humans raises profound ethical concerns that must be carefully considered. One of the most contentious issues is the potential for germline editing, which involves modifying the DNA of embryos, sperm, or eggs. Unlike somatic gene editing, which targets specific cells or tissues in an individual and does not affect future generations, germline editing results in heritable changes that are passed down to offspring. This raises concerns about unintended consequences for future generations and the potential for altering the human gene pool in unpredictable ways. Furthermore, the possibility of using CRISPR for genetic enhancement, rather than simply treating diseases, raises further ethical dilemmas. The prospect of designer babies with enhanced traits such as intelligence, athleticism, or physical appearance raises concerns about exacerbating social inequalities and creating a genetic divide between the enhanced and the unenhanced. To fully grasp the ethical complexities of CRISPR, it's crucial to understand the distinction between somatic and germline gene editing. Somatic gene editing targets specific cells or tissues within an individual, aiming to correct genetic defects or treat diseases in a targeted manner. For instance, scientists could use somatic gene editing to correct the genetic mutation that causes cystic fibrosis in lung cells, alleviating the symptoms of the disease without altering the individual's germline. Since somatic gene editing does not affect reproductive cells, these changes are not passed down to future generations. In contrast, germline gene editing involves modifying the DNA of embryos, sperm, or eggs, resulting in heritable changes that are passed down to offspring. This means that any genetic alterations made at the germline level would affect not only the individual whose genes were edited, but also their descendants. The ethical debate surrounding CRISPR hinges on the balance between its potential benefits and risks. On one hand, CRISPR offers the potential to alleviate human suffering by curing genetic diseases and developing new treatments for currently incurable conditions. The ability to eliminate genetic diseases from families and future generations is a tantalizing prospect that could revolutionize medicine. On the other hand, the long-term consequences of altering the human germline remain unknown. Unforeseen side effects, unintended mutations, and the potential for ecological disruption are just some of the risks that need careful consideration. Moreover, The possibility of unintended consequences for future generations raises profound ethical questions about our responsibility to future generations. Furthermore, CRISPR's potential for misuse is a significant concern. The technology's relative ease of use and affordability make it accessible to a wide range of actors, including those with malicious intent. The possibility of bioterrorism, the creation of genetically engineered bioweapons, and the potential for genetic discrimination are all scenarios that must be addressed. One of the most ethically fraught applications of CRISPR is the possibility of genetic enhancement, which involves using gene editing to enhance desirable traits rather than simply treating diseases. While the concept of enhancing human capabilities may seem appealing on the surface, it raises concerns about social justice, equity, and the very definition of what it means to be human. The prospect of designer babies with enhanced intelligence, athleticism, or physical appearance raises concerns about exacerbating existing social inequalities and creating a genetic divide between the enhanced and the unenhanced. Access to genetic enhancement technologies would likely be unequally distributed, further widening the gap between the privileged and the disadvantaged. Moreover, the pursuit of genetic perfection could have detrimental effects on human diversity and the appreciation of individual differences. The celebration of human imperfection and the acceptance of our limitations are fundamental aspects of what makes us human. Tampering with these aspects of our nature could have unintended consequences for our values and our understanding of ourselves. The ethical challenges posed by CRISPR are not confined by national borders. The global reach of this technology necessitates international collaboration and the establishment of global governance mechanisms to ensure its responsible use. International agreements and regulatory frameworks are crucial to prevent the misuse of CRISPR and to address the ethical concerns raised by its application in humans. The development of international guidelines for the ethical conduct of CRISPR research, the establishment of clear regulations for germline editing, and the creation of mechanisms for monitoring and enforcing compliance are essential steps in navigating the ethical maze of gene editing. A global effort is needed to ensure that this powerful technology is used responsibly and for the benefit of all humanity. To navigate the ethical complexities of CRISPR, we need to establish clear ethical frameworks and guiding principles for its responsible development and deployment. Principles such as beneficence, non-maleficence, justice, and autonomy should guide our decisions about the use of this powerful technology. Beneficence dictates that we should strive to use CRISPR for the benefit of humanity, prioritizing its application in treating diseases and alleviating human suffering. Non-maleficence requires us to minimize the potential harms associated with CRISPR, carefully considering the risks and unintended consequences of its use. Justice demands that we ensure equitable access to the benefits of CRISPR, preventing the exacerbation of existing social inequalities or the creation of new ones. Autonomy underscores the importance of individual choice and informed consent in decisions about the use of gene editing technologies. Public engagement is crucial in shaping the ethical and societal implications of CRISPR. Open and transparent dialogue between scientists, policymakers, ethicists, and the public is essential to ensure that this powerful technology is developed and deployed in a way that reflects societal values and priorities. Public forums, educational initiatives, and media coverage can help to increase public understanding of CRISPR, its potential benefits and risks, and the ethical considerations surrounding its use. Fostering informed dialogue and engaging the public in meaningful conversations about gene editing will be crucial in shaping the future of this transformative technology. The discovery of CRISPR-Cas9 has irrevocably altered the landscape of genetic engineering, presenting both unprecedented opportunities and profound ethical challenges. As we stand at the precipice of this genetic revolution, the decisions we make today will have far-reaching consequences for the future of humanity. The responsible development and deployment of CRISPR have the potential to revolutionize medicine, agriculture, and our understanding of life itself. However, the ethical complexities of gene editing, particularly germline editing, demand careful consideration and thoughtful deliberation. We must proceed with caution, guided by ethical principles and a commitment to the well-being of future generations. The ethical debate surrounding CRISPR is not merely an academic exercise but a call to action. It is a call for scientists to engage in responsible innovation, for policymakers to develop sound regulations, and for the public to become informed participants in this crucial conversation. By fostering open dialogue, promoting ethical guidelines, and engaging in thoughtful deliberation, we can harness the transformative power of CRISPR while mitigating its potential risks. The future of gene editing, and indeed the future of humanity, depends on our ability to navigate this ethical maze with wisdom, foresight, and a shared commitment to the common good. What are your thoughts on the ethical considerations of CRISPR? How do you envision the future of gene editing, and what role do you see yourself playing in shaping this transformative technology?